Okay, so in the last video, we did look at all the anatomy of the neuromuscular junction. And I also asked you to memorize or to remember two things. We have high concentration of free calcium in the extracellular fluid as compared to inside. Concentration of sodium is higher in the extracellular fluid as compared to inside. So remember these two. Okay, now what happens here? The first thing is we will look at the motor neuron, which is here. The motor neuron starts firing an action potential. So the action potential travels down the axon, comes to the axon terminal, right? And if you look into the blown up picture, this is the action potential that reaches the axon terminal. Listen carefully. Please hear me. As the action potential reaches the axon terminal, the voltage gated calcium channels open. So these channels open. And as these channels open, what comes in? Calcium comes in. So we have influx of calcium into the cell. Right? We have a lot of calcium now. Calcium causes the synaptic vesicles to fuse with the presynaptic membrane. So the synaptic vesicles fuse with the presynaptic membrane, and hold on. I cannot erase this, but it's okay. So calcium causes the synaptic vesicles to fuse with the presynaptic membrane and exocytose acetylcholine into the synaptic cleft. Now we have acetylcholine floating in the synaptic cleft. Now acetylcholine works as the ligand. So it's the key to the lock. The acetylcholine will then bind to the ligand-gated sodium channels, right? And as acetylcholine binds to the ligand-gated sodium channels, these gates open, and sodium, which is in high concentration, these are all sodium, okay? We have a lot of sodium in the synaptic cleft outside the cell. And as the ligand-gated sodium channels open up, sodium gets into the cell, into the muscle fiber. So sodium comes in. Good? Now remember, sodium carries positive charge. Now if a lot of sodium comes in, what happens to the polarity? Yes, sodium brings in a lot of positive charge. So we have a local depolarization at the motor end plate. So we have a local depolarization here. And this local depolarization is called end plate potential. So sodium brings in positive charge. There is local depolarization of the membrane at the motor end plate, leading to end plate potential. If the end plate potential is strong enough, that generates an action potential. So the end plate potential will generate an action potential, and the action potential will sweep all across the sarcolemma down the T-tubules. It'll go like this, the action potential, like this. So it'll sweep across the sarcolemma down the T-tubules. And as the action potential sweeps across the sarcolemma, that opens up voltage-gated sodium channels, and more sodium comes in. Good. So now we have so many sodium inside the cell, and the cell is now depolarized. Okay, now as the wave of depolarization comes down the T tubules, remember we have voltage sensitive calcium channels here. The voltage sensitive calcium channels, they, in response to the action potential, these voltage sensitive calcium channels, they undergo conformational change. So they change shape. And as they change shape, that opens up the calcium release channels. So these calcium release channels that are associated with the sarcoplasmic reticulum, the calcium release channels open up, and guess what? Calcium is released into the sarcoplasm. So now we have calcium coming out of the sarcoplasmic reticulum, and we have so many calcium floating in the sarcoplasm. Okay, so what will this calcium do? The calcium will go and bind to troponin. Do you remember I mentioned that calcium binds to troponin? Calcium will bind to troponin, and that will ultimately trigger or initiate muscle contraction or contraction of the sarcomere. So let's do this again. So here we have an action potential coming to the axon terminal. As the action potential reaches the terminal button or the axon terminal, these gates open up. What are these gates called? Yes, they are voltage gated calcium channels. They open and calcium comes in, right? Calcium causes these vesicles called synaptic vesicles, which are filled with acetylcholine. Calcium causes the synaptic vesicles to fuse with the presynaptic membrane and exocytose acetylcholine into the synaptic cleft. The acetylcholine binds to specific receptors or specific channels, and these channels are called ligand-gated sodium channels. They are called ligand-gated sodium channels. And as acetylcholine binds to the ligand-gated sodium channels, what comes in? The gates open and sodium comes in. Sodium brings in positive charge, which results in local depolarization of the membrane, or to be precise, we will say that as sodium comes in, there is local depolarization of the motor end plate, leading to a state called end plate potential. End plate potential. If the motor end plate area is depolarized enough, the end plate potential gives rise to an action potential that sweeps across the sarcolemma down the T tubules, and thereby the action potential sweeps all over the sarcolemma and also down the T tubules. As the action potential sweeps across the sarcolemma, we have these channels, and these are called voltage gated sodium channels. They open up and more sodium comes in. Now on the T tubules, we have voltage sensitive channels or voltage sensitive calcium channels. Due to the action potential passing along the T tubules, the voltage sensitive calcium channels change conformation, leading to opening of the calcium release channels associated with the sarcoplasmic reticulum, and that's why a lot of calcium are released from the sarcoplasmic reticulum into the sarcoplasm. This calcium will go and bind to troponin and cause muscle contraction. Okay, now we will look at the PowerPoint and make sure we understand everything before we move into the physiology of contraction. Alright, so looking at a neuromuscular junction, we do see the motor terminal axon, so terminal axon of the motor neuron. We see an action potential or a nerve impulse in yellow. We do see voltage-gated calcium channels. We also see synaptic vesicles. These are the synaptic vesicles filled with acetylcholine, denoted by the red balls. We also see the sarcolemma of the muscle fiber. We see ligand-gated acetylcholine receptors. In here, they are called nicotinic acetylcholine receptor. They're the same thing as ligand-gated sodium channels. We also see the sarcoplasmic reticulum in red here. And we see the DHP receptors. Do you see the DHP receptors? These are actually the voltage sensitive channels. And then the rhinodine, the RYR receptors. These are the calcium release channels. They are popularly known as RYR channels or rhinodine receptors or rhinodine channels. Alright, so first what's happening? We have an action potential that reaches the axon terminal and voltage-gated calcium channels open. As the voltage-gated calcium channels open, calcium enters the cell, as shown by the purple balls here, and calcium causes the synaptic vesicles to fuse with the presynaptic membrane and exocytose acetylcholine into the synaptic cleft. Now you have acetylcholine, denoted by the red balls, in the synaptic cleft. The acetylcholine binds to the ligand-gated sodium channels, which are shown in blue on the sarcolemma, and opens up those gates. And therefore, sodium enters the muscle fiber. Sodium brings in positive charge into the muscle fiber and causes the fiber to depolarize, or it forms a end plate potential. That's a local depolarization at the motor end plate. And if the depolarization is strong enough, it triggers an action potential that sweeps across the sarcolemma, as shown by the black arrows, across the sarcolemma down the T tubules. As the action potential moves down the T tubules, there are voltage sensitive receptors called dihydropyridine receptors or DHP receptors that change shape or conformation, resulting in opening up of the calcium ion release channels, also known as rhinodine receptors or RYR receptors, shown in light blue here, that are on the sarcoplasmic reticulum. Therefore, I repeat, as the action potential moves down the T tubules, the DHP receptors change shape or conformation that causes the RYR receptors or the calcium ion release channels to open, and calcium is released into the sarcoplasm. So we have a lot of calcium in the sarcoplasm now. The release of calcium into the cytoplasm, or sarcoplasm in this case, raises the sarcoplasmic calcium ion concentration, thereby stimulating muscle contraction. The process by which action potential causes contraction is termed excitation-contraction coupling. Alright, so I'll stop here and then start with this slide in my next video.